week of physiology course. So in this week, we will be talking about the three lectures like lives of mammals in frigid places. We will talk about the neurons, and also we will talk about the sensory processes. So talking about the lives of mammals in frigid places, we will take a case study of reindeers that are the Rangifer tarandus. That's their zoological name. So these reindeers, they occur in Siberia and other far northern regions, typically give birth in May when the ground remains snow-covered and the air often cools to below 0 degrees centigrade overnight. So at the moment of its birth, a reindeer calf experiences a drop in its environmental temperature. Usually that's like 37 degrees centigrade inside its mother's uterus to the prevailing air temperature. So if their temperature is minus 3 degrees centigrade, the calf's environment temperature plummets by 40 degrees centigrade at birth. So the air sometimes is much colder, and some cows they experience a drop of like 50 to 60 degrees centigrade when they are born. So these newborns are wet, covered with amniotic fluid, and a strong wind may blow. So reindeer calves must thermoregulate on their own from the moment they are born. So they have to regulate their temperature because they do not huddle with each other or with the adults, or they do not have to nest to protect them. So they are perhaps the most precocial, or we can say the adult-like of all the newborns. So what they have to do, they have to thermoregulate that temperature. So they stand the moment they are born when 2 days old. So they can run faster than a person because herds typically move from one place to from place to place instantly to find food. So the newborn calf has no luxury of resting. So this newborn reindeer calf must thermoregulate. That's what we are going to discuss here. So although mother reindeer provide milk to their young, but they do not keep them warm, and the Arctic environment where birth occurs may be very cold. So how these animals they do this? How do they thermoregulate the temperature? How do they adapt to that climate? Even if they are mothers, they don't help them in that. So they just behave like, so they have, what they have to do is to thermoregulate that temperature. So regarding the food, nutrition, energy metabolism, and thermoregulation in the lives of adult reindeer, we all know that reindeer as a species are probably the most adapted of all inland mammals to cold exposure. So some herds live year-round in places where the average annual temperature is like minus 5 degrees centigrade. So an intriguing aspect of animal species that are extremely well suited physiologically to live in stressful habitats is that their very presence can permit other species to exist there as well. For example, in deserts, the existence of rodents that have evolved extremely low requirements for ingested water permits many water-dependent predators to exist as well. The rodents provide the watery food the predators need. So similarly, in the far north, the existence of reindeer permits the existence there of wolves and their predators. So reindeers, they have many characteristics that help them prosper in the far north. So although adult reindeer weigh an average about 1/3 as much as old moose, the reindeer with the ground is about the same as that of moose. So adult reindeers in their winter sorry. So these adult reindeers in their winter village have lower critical temperatures of like minus 30 degrees centigrade, or we can say lower in still air. Thus we can say the air temperature, it doesn't drop far below their lower critical temperature, and reindeer in their winter village in still air never have to increase their metabolic rates by much above basal, like you can see here in this figure. So as an amusing comparison, for example, we have the Laplanders and Norwegians, they are equipped just with insulation nature, which gave them like, for example, the neck, they have lower critical temperatures of plus 24 degrees centigrade or minus 27 degrees centigrade. So only by dressing in something like a radial pelt can a person have a lower critical temperature that's compatible with the Arctic life. So one reason that might be for the low energy costs of adult reindeer in the frigid air of winter is their pelage. So a dense underfur of fine hairs that thickly covers their skin. So this pelage is actually that protects them from the outside cold temperature. So the dense underfur of fine hairs that thickly covers their skin and the longer guard hairs of the pelage which protrude beyond the underfur and hang over its outer surface are filled with many air spaces like microscopic bubble wraps, like here you can see in this picture. So the air inside these hairs is actually motionless, meaning that each hair in itself has a high insulative value. So in common with other large species of mammals, reindeer undergo the dramatic seasonal molt. So in winter, a reindeer's fur is like 3 to 4 centimeters thick over much of its body. For example, here you can see. So these are the values of the reindeer's fur, the diameter of the reindeer's fur in winter and summer. So like I said, in winter, for example, we can say that the dramatic in winter, the reindeer's fur is like 3 to 4 centimeters thick over much of its body. So all body surface of a reindeer, even the nose, have a hair covering. So the Homo erectus adaptation exists from one end of a heterothermic leg to the other, while the lipids in the upper leg of a reindeer or cow have chemical structures that give them a gel-like structure at 37 degrees centigrade and cause them to become hard at 0 degrees centigrade. So lipids from the outer extremities have different chemical structures that give them a gel-like or oily texture near 0 degrees centigrade. So these are the different adaptations that the reindeers make because of their cold temperature, so that how they can adopt, or we can say the how they can do this thermoregulation because of the pelage, because of these chemicals that are secreted in between the legs. So one way to examine the spatial diversity of chemical structures is to look at the abundance of key diagnostic fatty acids in the marrow lipids of limb bones. So as seen in figure here, you can see. So this graph here, it shows the oleic acid that contains one double bond and therefore is a monosaturated fatty acid. And these here are the foreleg and hind leg, the blue and these and the black dots. So oleic acid, which is an unsaturated fatty acid, becomes dramatically more abundant as a constituent of marrow lipids as one moves out along a leg, whereas palmitic acid and stearic acid, that's here in this graph, the palmitic acid and stearic acid, they are fully saturated fatty acids, and these saturated fatty acids, actually what they do is that they become a constituent of marrow lipids. So in this way, the bone marrow lipids in the legs of reindeer are reminiscent of the brain phospholipids of fish from various classes. The same basic trends exist even in the legs of many tropical mammals. So thus, although the trends are significant for reindeer, but they do not seem to be specific adaptations to a truly frigid climate. So food and nutrition, they also are great challenges in the environments where the reindeers live, the cold environment. So like other deers, the reindeer are ruminants. So thus rumen processes as well as digestive and absorptive processes play vital roles in their physiology of food and nutrition. So in key diversity of plants, for example, we have the 37 genera that were found in the rumens of manhood. So they obtain an exceptional degree of nutritional benefits from eating species of lichens and a variety of other species of far northern plants that are not much eaten by other mammals. So this is how they actually adopted this climate regarding their food and nutrition. So we can also say that the as like in this picture you can see the reindeers often depend heavily on lichens for food in the winter. So this has been known for many years from visual observations of their feeding and has recently been confirmed by using certain genomic methods. So by analyzing the DNA in reindeer fecal pellets and applying DNA barcoding methods, the investigators or researchers, they have obtained impressively detailed information on the foods that the reindeers consume. So regarding them, the reindeers, they consume different kinds of foods. For example, herbs, grasses, trees, mosses, lichens, or terrestrial lichens. But the main food that they eat is the lichens. So 80% of the food, when the DNA of the reindeers was analyzed by different researchers, they found that lichens was the main component of their food. So in winter, the lichens may account for, like you can see here, the 80% of the diet. So lichens are often the salvation of reindeer in winter in terms of supplying energy because they are abundant, and as we have already said that reindeers can have a high proportion of their nutrient value. So however, the lichens are far from being nutritionally complete because they are low in protein and low in minerals such as sodium. Like here you can see. So here in this graph you can see the lichens. They are the major component of reindeer's winter diet in Finland, which are low in both protein and minerals. So in this figure you can see that it summarizes actually the composition of the foods that are available to reindeers throughout the year in Finland. That's well worth close study because it illustrates dramatically that animals in the wild, away from veterinarians and manufactured feeds, often face substantial nutritional stresses. So during winter, the lichens and vascular plants that dominate the diets of reindeer, they are collectively low in protein, low in multiple minerals, low in highly digestible carbohydrates, but they are high in cellulose and hemicellulose. So here in this table, you can see that reindeers and the microbial symbionts in their rumens make adjustments as the season change. For instance, certain strains of the reindeer, they fatten dramatically as winter approaches, so thereby reducing their need for winter food. So in addition, the community of the community of the microbes in the rumen, it changes in composition in ways that respond to the shifts in the type of foods that they eat. Here in this table, you can see that it presents one example indicating that the microbes that digest woody, fibrous plant material, including the cellulose and hemicellulose, they increase in winter. So when the reindeer ingest considerable amount of such material, a recent study has shown that mixed rumen microbial community in Norway reindeer become 4 to 6 times more capable of breaking down these lichens when the lichens were chronically in the diet than when the lichens were chronically not eaten. So here you can see that the percentage of microbes that can carry out each activity in summer and winter. For example, for the fiber digestion, it was like 31%, and then in winter, it increased up to 74%. The cellulose digestion was 15 and it increased to 35, and accordingly for the hemicellulose and proteolysis or starch digestion. So what they are doing, they actually are adapting that how they can digest different kinds of foods that are rich in fiber, cellulose, hemicellulose, or proteins or starch. So in addition to the physiological and anatomical features, we have already discussed the distinctive feeding behaviors and migratory behavior of reindeer herds. They also play a key role in their successes. So in the far north, food is thinly distributed even when for animals that are on a diversity of plants. So talking about the newborn reindeer, when the reindeer are born, they already have a well-developed pelage that consists of woolly, hollow hairs. So their fur actually provides substantial insulation against the cold temperatures. Or we can say as soon as the uterine fluids have evaporated away and the hairs are dry. So from the moment of birth, the reindeer exhibit a typical homothermic relation between their metabolic rate and the air temperature. As you can see here by the red line here, the 1 day old. So it actually makes a link, makes it synchronize the air temperature with the resting metabolic rate. So this red line here indicates the 1 day old, how it synchronizes its air temperature with the metabolic rate. So this we may call as thermoregulation. So newborns are able to raise their rate of metabolic heat production to at least twice their resting rate. So by virtue of combined effects of their pelage insulation and thermogenic ability, so by virtue of the combined effects of their pelage insulation and this thermogenic ability, newborns are able to keep their body temperatures at 39 to 40 degrees centigrade. And when the air is like minus 20 degrees centigrade to 25 degrees centigrade. So for at least a few hours in still air. So this performance probably represents the pinnacle or near pinnacle of thermoregulatory ability among all terrestrial newborns on Earth. So that means it remains true that reindeers are born into an environment that can be very harsh, and the thermoregulatory abilities of newborns are far inferior to those of adults. So many newborns, they die even if they get wet from precipitation or if the wind blows briskly or the air temperature remains very low for a day or more. So microscopic studies performed in about 1980 indicated that most of the major deposits of adipose tissue in the body of a newborn reindeer are the brown fat. So nonetheless, the physiologists, they assume that the function, not just the morphology, must ultimately be directly studied for function to be understood. So as we have discussed in the previous chapter, that thermogenesis by brown fat is ordinarily actuated by the sympathetic nervous system secreting the norepinephrine. So on the basis of this fact, an animal's response to a non-epinephrine injection has been found used as an informative. So when newborn reindeer are injected subcutaneously with a standard dose of this epinephrine injection, they respond with a large increase in their rate of oxygen consumption. So older animals, they actually exhibit less of a response, indicating that the capability for non-shivering thermogenesis by brown fat decreases as the reindeer ages. But when 1 day old reindeer is given this norepinephrine injection subcutaneously, they exhibit a large percentage increase in the rate of oxygen consumption, indicating a well-developed capability for non-shivering thermogenesis by the brown fat. So reindeer newborns are not alone, alone in being well endowed with brown fat. In fact, most placental mammals, including humans, like here in this figure, you see that have extensive masses of brown fat at birth. So pigs are the exceptions in this. So in large-bodied species such as reindeer and humans, the brown fat of newborns typically declines. So what we have seen in this is that in large-bodied species such as reindeers and humans, the brown fat of newborns typically declines rapidly with age. So this decline is actually, we may say that it's particularly rapid in the ruminants. So we can say that in reindeers, sheep, goats, and other species of ruminants that have been studied, all the brown fat that's present in the newborn, they undergo uh, a genetically programmed decline fast enough that there is no brown fat by 1 month of age. So this is demonstrated by the fact that in 1 month old animals, the gene for UCP1 is no longer expressed and no tissue reaction with this specific antibody occurs. So simultaneously, we can say the metabolic response of the young animals to a norepinephrine injection declines substantially. So another interesting question is how the large masses of brown fat in unborn near-term young are regulated so that they do not catabolize large amounts of the young foodstuff. So we can see here that logic suggests that prior to the birth, a young animal will profit most from the foodstuffs it has by using them to grow and mature. So brown fat, if uncontrolled, could turn large quantities of foodstuffs into heat and chemical wastes. So some of the experiments have been done that reveal that if near-term sheep fetuses are cooled inside the uterus of their mothers, like here you can see in this picture, so that's a sheep that's near the term. So what happens? They do not activate their brown fat or engage in these processes. However, if the blood flow from the placenta of a near-term fetus, here you can see the placenta, the blood flow in the umbilical cord between the fetus and placenta, it could be blocked or cut off by the severing of umbilical cord, and the brown fat is then able to respond vigorously to the chilly outside environment by producing the heat. So a similar process may occur in reindeer. Future research has been done on that. So here you can see the ventilators. The lungs were ventilated with air and the fetus rate of oxygen consumption from the air could be measured. So this was actually the experiment that was carried out. So moving to the hibernation as a winter strategy, new directions and discoveries have been made regarding that, how the hibernation occurs in these frigid places. The hibernating species of placental mammals, they are coded for retaining brown fat as adults. So there was a scientist, Conrad Gessner. He first described this brown fat in 1551. So he was studying the adults of hibernating species, the European mammoth. For 4 centuries thereafter, brown fat was observed in the adults of one hibernating species after another. So the association of this brown fat with the hibernation was so consistent that although the function of brown fat was unknown, the tissue was dubbed the hibernation gland. So today we know a great deal about the function of this brown fat in adult hibernators. So first and foremost, brown fat is thermogenetic tissue that takes the lead in rewarming during arousal. So emergence from hibernation, we can say. So as we discuss the hibernators, further two species will receive special attention. One is the Arctic ground squirrel and the alpine marmot. So ground squirrel, which weighs about 500 to 800 grams, is found widely in the far north, Alaska, the Yukon, and the neighboring parts of Canada. The marmot, which is larger and weighs about 4 to 5 kg, occurs high in Europe and Alps. So both species employ hibernation as a strategy actually to cope with the stresses of the winter in some Earth's most frigid places. So great majority of these hibernators, they have adult body size of like 5 kg or less, we can say. So one reason is probably that the energy savings they are afforded by hibernation decline with the body size. As shown in figure here, you can see the weight specific. So here on the x-axis, we have the body weight on scale, and this is the weight specific metabolic rate. So the weight specific metabolic rates of hibernators in hibernation, that's in the red line here, you can see, are statistically the same regardless of their body size. So it's remaining constant, the metabolic rate, the weight specific metabolic rate. So thus the energy savings achieved by hibernation decreases as body size increases. So in the fact of this, however, you can see the animals that are not hibernating animals. So this is in case of hibernating animals, the red line here, the metabolic rate, the weight specific metabolic rate remains constant. However, in case of blue line here, which is the non-hibernating mammals, they actually, the small bodied species, we can say, they have higher weight specific metabolic rates. So it's increasing here, the non-hibernating mammals. So we can say the energy savings achieved by hibernation decreases as the body size increases. So in fact of this allometric trend, natural selection seems not to have favored the evolution of hibernation. So as we discussed the Arctic ground squirrels, alpine marmots, and other hibernates, it's worth reflecting on the fact that much of we know about them today is consequence of actually the technological revolution, and that has occurred in the last 50 years. So with these advances in technology, records like that, and you can see here in figure, they have become possible and have revolutionized knowledge of physiology of hibernation. So the record in the figure shows that the body temperature of a free-living and undisturbed Arctic ground squirrel for 8 continuous months, from September to May, the full duration of its hibernation season. So the body temperature was recorded with a data logger. The soil temperature in this ground squirrel hibernation is also shown in this, which is about 10 to 15. So what the Arctic ground squirrels, how they supercool during hibernation and arouse periodically throughout their hibernation season. So a striking aspect of the record in in the previous figure that we have shown is that by early December, the ground squirrel's body temperature fell below 0 degrees centigrade during periods of hypothermia. So the soil temperature fell as winter progressed, and as the soil temperature fell, the ground squirrel maintained a large difference between its body temperature during hypothermia and the soil temperature. So the body temperature fell below 0 degrees centigrade, a very unusual state for hibernators. So we can say the freezing point of the body fluids of Arctic ground squirrels remains the same during winter as it is in summer. Yet the Arctic ground squirrels sometimes cool during hibernation to body temperature of -2 degrees centigrade to 3. So they actually do not freeze at these times. Instead, they super cool. So just as some uh, poikilotherms do in winter. So to be the endothermic in this way, the hibernating Arctic ground squirrels must increase their rate of heat production in midwinter to rates above those that are seen at other times. For example, September and October, when the soil is warmer, they can simply let their body temperature approximate the soil temperature. So what happens, the rate of heat production of these hibernating ground squirrels is increased as needed by the brown fat thermogenesis. So as we have already studied that it's common for mammals and birds that are in states of controlled hyperthermia to prevent their body temperature from falling lower than certain levels by elevating the metabolic heat. So composition of the lipid consumed before hibernation, it also affects the dynamics of the dynamics of hibernation. So composition of foods that animals eat is often as significant as the amount of food. So dramatic illustration of interaction between the food consumption, that what kind of food they are eating, and physiology. It has emerged in recent years from studies of these hibernators. So storage lipids of hibernators and other mammals, they reflect in their composition the suites of fatty acids that are present in the foods that animals eat during fattening. So each triglyceride molecule is built from these fatty acid molecules. So fatty acids also, they are termed as the saturated fatty acids. They're also termed as SFAs, contain no carbon, the carbon double bonds. We have got some monosaturated fatty acids, or they contain one carbon double bond per molecule. Finally, we have the polysaturated fatty acids that contain two or more double bonds per molecule. So we have already discussed in our previous chapter that the mammals are incapable of synthesizing most of the polysaturated fatty acids from starch. However, if they eat plants that are rich in these polysaturated fatty acids, they can use those directly or employ them as substrates for the synthesis of these polysaturated fatty. So like here in this picture, you can see particular fatty acids shown are the linoleic acid, or we can see omega-3 and linoleic acid, omega-6. These are the simplest precursors of the two types of polysaturated fatty acids. Because their structures and conformations, they are different. The two types of these polysaturated fatty acids, when incorporated into the membrane phospholipids, have different effects on the structure of the lipid matrix and also create a different context to the function of membrane proteins. So we have here the table, the hibernation performance in chipmunks that are fed three diets. That's the polysaturated rich diet, intermediate, and then we have saturated fatty acid rich diet. So the lowest body temperature that did not provoke the arousal was like 0.6 when it was fed with the polysaturated fatty acids. Rate of consumption at an ambient temperature was like 0.034, and length of each continuous hibernation was about ambient temperature of 5 degrees centigrade. So what this table actually depicts is that hibernation actually is correlated with the type of diet that the animals they are eating. So although we have the periodic arousals that detract from the energy savings of these hibernation, their function is unknown. So a qualitatively simple pattern is exhibited by other species. For example, we have the alpine marmots, for example, the reap great energy rewards by entering the hibernation each winter. You can see here in this picture. So this blue line here, it indicates the annual cycle of energy used if the marmots did not hibernate. And this is the actual annual cycle of energy used. So winter energy used if there were no periodic arousals. So this is the blue line here. So even more could be saved if no periodic arousals occur. So given that animals pay an energy price for periodic arousals, it seems that arousals must have important functions. So in the early days of research on this topic, a living hypothesis for periodic arousals was that they allow hibernators to void the wastes. So talking about the intersection of sociobiology and hibernation physiology, so recent analysis of data from many species have established that hibernation is correlated with increased survival in the wild. For example, one question of interest is that whether individuals capable of hibernation they enjoy the lower odds of death during months when they are hibernating than when they are not. So in this regard, the available data that provide an emphatic answer is on average in species of hibernating small animals, an individual is about 5 times more likely to die in a month when it is active rather than when it's hibernating. So hibernation probably has this effect mostly because of avoidance of predators. Hibernating animals, they remain always in their hibernacula instead of moving around in large environment where predators can spot them. So avoidance of predation may in fact have been a significant factor in the evolution of hibernation. So because of its effect on odds of survival, hibernation can actually affect an animal's odds of reproduction. So there are certain male ground squirrels that illustrate another consideration that arises in some cases at the interface of hibernation biology and reproductive biology. So in ground squirrel species that have been studied, the testicles are regressed during hibernation. From what is known, no growth is possible. So while hypothermia continues, as you can see here in this figure, it shows that this phenomenon in male golden mantle ground squirrels. So testicular development starts only after high body temperatures have been restored following the end of hibernation. And because full development requires several weeks, males are unable to breed until long after they have terminated hibernation. So here in this figure, you can see that by statistical calculations, an index of synchrony can be calculated for arousals in groups of marmots. So a high index for a hibernating group means that the individuals monitored with radio telemeters in the group aroused highly synchronously. So based on the data for 14 hibernating these animals, groups of alpine marmots, the figure here shows the relationship between the loss of body weight and the synchrony. So as you can see, the individuals in highly synchronized groups lost approximately 20 to 25 degrees centigrade of their body weights during the winter hibernating season, but individuals in some poorly synchronized groups lost about 40 to 45%, roughly twice as much. So because relatively great loss of body weight lowers animals' likelihood of survival, we can say that this analysis reveals that synchrony during periodic arousals has important survival consequences. So that was all about the mammals, how they regulate their body temperatures and other body parts regarding their food and nutrition and the temperatures, how they adapt themselves to that frigid places. So moving on to our chapter 12, that's about the neurons. So we will be talking about how the sensory processes, or we can say the neurons, they help in different physiological functions of the body of these animals. So the main thought points of this chapter will be that we will discover that the electrical basis of neuronal functions, like ability of neurons to generate electrical signals and how they propagate through large distances. We will also talk about the physiological control processes and the contrasting functions of neuronal and hormonal modes of integration. So let's start with a case study of a squid. So we all know that locomotion, or we can say the movement in a squid, whether for capturing a meal or to avoid becoming one, depends on jet propulsion. So contraction of muscles in the squid's outer mantle expels the seawater through a movable siphon, propelling the animal in the opposite direction. So it's true in all animals that feeding escape and similar behaviors in the squid are controlled by the nervous system signals, which travel rapidly in a point-to-point manner, or we can say from one specific cell to another. So these signals actually arise from the properties of the nerve cells, which are termed as neurons. So they have long been like cable-like processes, which are termed as axons, that convey these electrical signals rapidly and faithfully from place to place in the body, even over long distances. So in the squid, the sensory neurons, such as those in the eyes, encode information about the squid's environment and convey these signals to the brain. So there the signals that are integrated into a decision to attack or retreat. So the brain then sends command to the mental muscles in part through a set of large neurons with large rapidly conducting those axons. So this is how they convey information, how they can protect themselves from the enemies. So in this chapter, we will discuss that how the squid giant axons have played an important role in our understanding of these neuronal functions. So diameter of these giant axons can be as large as like 1 millimeter, or we can say 1,000 micrometers. And for more than a half century, investigators have taken advantage of this cellular size to perform experiments on these squids that have revealed the mechanism of these neuronal signaling. So the physiology of control, that means the neurons and the endocrine cells, how they are compared. So starting with that, we all know that animal needs to function like a coherent organism, not like a loose collection of cells or intracellular mechanism. Or we can say that integration, which is a general term that refers to the processes such as summation or coordination that produce coherency and result in harmonious functions. So this cellular integration refers to the processes within the cells. So whole animal integration refers to the selective combination and processing of these sensory, endocrine, or central nervous system. In short, we can say the CNS information in ways that promote the harmonious functions. So nervous and endocrine system are also described as control systems because the nerve cells actually and the endocrine cells control the ways in which the other cells function. So this use of the concept of control systems is complementary. So to see the relations, for example, consider a control system of objects such as cars, or we can say the computers, where the physical entities that implement control functions are made of materials such as copper, silicon. So in animal, control functions are mostly carried out by these nerve cells and endocrine cells. Like the other functions, I already gave you the example of cars or computers, where these kind of materials such as copper and silicon, they behave in the same way like the nerve cells and endocrine cells behave in the animal body. So the nervous system and the endocrine system work in systematically different ways to control and coordinate the cells of an animal. So like here in this figure, you can see that it shows a signal in a neuron that travels electrically. So this signal actually travels electrically all the way to its target cell. So transmission along the cell process is very fast and especially highly defined. So when the electrical signal arrives at the end of neuron processes, it causes the release of a chemical substance, a neurotransmitter that diffuses quickly across the minute gap between the neuron processes and the target cell. So when this chemical substance arrives at the target cell, what happens is that it binds with the specific receptor molecule, but that binding is a non-covalent binding on the cell, activating the target cell response. While in this figure B here, you can see, in contrast to this figure, it shows that when an endocrine cell emits a signal, so endocrine cell here, it emits a signal. It does so by secreting a chemical substance termed as the hormone into the general body circulation. So signal travels more slowly than a neuronal signal because it's carried by blood flow instead of being specially highly circumscribed. So the signal is transmitted to all the cells in the body, the target cell, the cells that respond, or the subset of the cells. So how these neurons transmit these electrical signals to the target cells? So they have actually 4 functional regions that typically correspond to their 4 major structural regions. So because neurons are commonly likened to the wires in the type, for example, we have a telephone or we have computer networks, so neurons behave like the wires. So most people have an intuition understanding that what these cells do. So a neuron is a cell that's adapted to generate an electrical signal in the form of a brief self-propagating impulse, which is called the active potential, that travels from place to place. As we have already here in this figure, it reveals that a neuron has 4 parts: the dendrites, cell body, axon, and presynaptic terminals. So these are the 4 parts that typically respond to their 4 structural regions. So the 4 functions that these 4 parts perform is the input, integration, conduction, and output as a controller cell within the animal's body. So when a neuron receives the input signal from other neurons or other sensory cells, we can call it the synapses. Usually the synaptic input occurs along branching processes known as these dendrites. So although synapses may occur in the cell body as well, these impulses that are arriving at a synapse from a presynaptic cell cause the release of a chemical substance which is called a neurotransmitter into the synaptic clip. The cell body, also called the soma, is commonly, here you can see the cell body, which is also called as the soma, is commonly the part of a neuron where the signal integration and impulse generation occurs. So this long slender body, which is called the axon here, is the conduction component of a neuron serving to propagate action potentials along its length. And these presynaptic terminals here, it usually constitutes the places where neuronal output occurs or other types of cells such as muscle fibers. So an action potential arriving at this presynaptic terminal here So at this presynaptic terminal, the output of the neuron, that's the secretion of neurotransmitter, can alter the activities of the other cells, we can say. So an action potential arriving at this presynaptic terminal triggers the release of molecules of this neurotransmitter across the synapses and exert a specific physiological effect on the target cell. So these neurons that form synaptic ends on the cell are said to be innervate that cell. So we have here the neural circuit mediating the startle response in the cockroach, that's the Periplaneta americana. So what actually happens is that these nervous system and endocrine systems, they tend to control different processes. So neural lines of communication are capable of much finer control, both the temporal and spatial. So whereas the nervous system controls predominantly the fine rapid movements of discrete muscles, what endocrine system does, it typically controls more widespread prolonged activities such as the metabolic changes. Let's consider an example of running to catch a fly ball in baseball. So it requires rapid computation and very specific control of these discrete muscles in split-second time that functions that can be mediated only by the nervous system. So in contrast, the control of metabolism or growth requires the modulation of the many tissues over a protected period. So most tissues in animals' body are under the dual control of nervous and endocrine system. So skeletal muscle illustrates the relationship of this dual control. So how these neurons are organized into functional circuits in the nervous system. The functions of a nervous system actually depend on wiring, the anatomical organization by which these neurons are connected into circuits. So any behavioral activity, for example, swimming in the squid, with which we open our chapter, is a property of neural circuit that mediates it. So we have here one more example. Suppose we walk into the kitchen and surprise a cockroach. The cockroach jumps, exhibiting a startle response in which it turns away from the disturbance and prepares to run. So this simple behavior of a cockroach act is mediated by actually the electrical signals and chemical synapses within the cockroach's nervous system. So the cockroach's jump is actually the reflex behavior response to a distinct stimulus. So air currents or airborne sound waves vibrate filiform hairs that act as wind receptors at the cockroach's posterior end, like here you can see in the picture. So what actually happens? So there are the stimulus, the nerve impulses, and the response. So we have 5 responses. So number 1 is the stimulus, that is the air puffy, and the other is the sensory neuron, that's wind receptor, and third is the giant intraneuron, and then we have the cockroach's leg motor neurons here, the motor neurons that activate the leg muscles, and then the muscle tension here. So these motor neurons in turn, they activate these leg muscles actually. So we have here the melanocytic ganglion at the intraneurons that synaptically excite these leg motor neurons, and finally we have the motor neurons in turn that activate the leg muscles. So this is what happens actually when we, for example, I already talked about, for example, we enter the kitchen and how that cockroach jumps. So these are the different sensory things that are happening. So number 1 is the sound waves or air currents that vibrate the hairs of this cockroach. And number 2 is the vibration of the hair generate these nerve impulses in sensory neurons. And then number 3 is the sensory neurons, they synapse with and excite the giant intraneurons that interiorly in the ventral nerve cord. And then at the melanocytic ganglion, the intraneurons synaptically excite these leg motor neurons that makes the cockroach to jump. So this is what actually happens. So cellular structure of a vertebrate neuron, we will talk about this. So neurons, as we have already seen earlier, are cells that are generating electrical impulses and transmitting those impulses from place to place. So they have long processes which relate to their functions, acting, for example, as the conduits for long distance transmission. The cytology of a neuron neuronal soma is broadly similar to that of non-neuronal cells. So a neuronal soma contains a nucleus. Here you can see, or we can see the cell body. That's the this soma. So it consists of nucleus and most of the organelles and cytoskeletal elements. For example, the mitochondrion, Golgi apparatus. It has smooth endoplasmic reticulum, rough endoplasmic reticulum, microtubules. It has neurofilaments and actin microfilaments. So neurons are very active in protein synthesis and have tend to appear in light microscopy as nasal substance. So these neurons, they can be classified according to the number of processes that emanate from the soma. So neurons may be unipolar, having one process, or they may be bipolar, having two processes, or multipolar, having performing three or more processes. So unipolar neurons, they predominate in the central nervous system of most invertebrates. Multipolar neurons, they are present in the vertebrates central nervous system, and many sensory neurons are bipolar in various taxa. So neuronal responses exhibit a geometric variety and complexity, we can say. So like here in this picture, you can see the dendrites of spinal motor neurons. They are short and branch repeatedly into the repeatedly into so these dendrites of most neurons have continuously varying diameters and they lack myelin sheaths. So in general, we can say that the broader dendrite trunk assemble the soma in fine structures. So they contain rough endoplasmic reticulum, mitochondrion, microtubules, neurofilaments, and occasional Golgi apparatus. So axon of a neuron is single and long with a relatively constant diameter and few collateral branches. So these the larger vertebrate axons are surrounded by the myelin sheath. So axons generally lack the rough endoplasmic reticulum and Golgi apparatus. So axon, functionally, we can say axon is usually the portion of the neuron that supports the action potentials, or which we can say which propagate or conduct along the axon without decrement, carrying the information from the cell body to the axon terminals. So let's move to the glial cells, how they support the neurons physically and metabolically. So cells that are referred to collectively as glial cells or neurological cells surround the neurons, like here you can see in this figure. So these were discovered by Rudolf Virchow in 1821 and named the neuroglial cells in 1846, and thought that their primary function was to bind the neurons together and maintain the form and structure organization of the nervous system. So we have different types of glial cells. They play diverse roles in nervous system. So vertebrate nervous system have two kinds of enchything glial cells, for example, Schwann cells and oligodendrites. So other glial cells, also called as astrocytes, they actually line the outside surface of the capillaries in the vertebrate central nervous system. So simple electrical circuit. So let's move to the ionic basis of a membrane potential, how it works. So simple electrical circuit of a flashlight. Let's take the example of that. So what are the properties of electrical signals of neurons and how these signals are generated? Let's begin with a brief review of basic electrical concept. For example, we have the protons and neutrons that are electrically charged. So protons are positively charged and electrons are negatively charged, and we also have ions that are the atoms or molecules that appear in net charge. So because they have equal number of protons and electrons, the fluid it contributes to so they have unequal number of protons and electrons. So the net movement of charges constitutes an electric current, which is analogous to the hydraulic current of fluids. flowing in a system of pipes. The separation of positive and negative electrical charge constitute a voltage or electrical potential difference. So this potential difference can do work when the charges are allowed to flow as current. So voltage is analogous to a height difference, or we can say it's head of pressure in a hydraulic system. So like here in the figure, you can see the simple electrical circuit of a flashlight. A battery that provides a voltage closing the switch allows current to flow through the electrical circuit. The electrical circuit in the flashlight is actually the flow of these free electrons along the metal wires. So current flows through the light bulb filament, which acts as a resistant that limits the current flow. So electrical circuit in cells, they are actually similar to the circuit in a flashlight because they differ in some important ways. So in cells, both the inside and outside or media are aqueous solutions in which the electrical charge are ions rather than free electrons. So furthermore, we can see that all currents in the cells are carried by ions, and any voltage or potential difference results from local imbalance of these ionic charges. So we already had gone through that in our chapter 5, that fluids rather than a few nanometers from a membrane are electrically neutral with equal number of positive and negative charge. So because of this charge neutrality, we can say of the bulk solution, the only portion of the cell that directly determines the electrical properties in its outer limiting cell membrane. So these cell membranes, they have actually the passive electrical properties, that's the resistance and capacitance. So all cells respond to these electric currents, but not all cells generate these action potentials, or we can say the nerve impulses. So the universal response are passive in response to that, but action potentials are active responses in which the properties do change. So we can say the cell's passive response depend upon the passive electrical properties. So membrane actually exhibits the capacitance that can be measured in farads because of the insulating properties of the bilayer. In electrical circuit, a capacitor has two conducting plates that are separated by an insulating layer in cells. Like here you can see. So resistance and capacitance of these depend on the membrane area, the specific membrane resistance and capacitance, which are measured per unit of area. So let's relate this example to a squid giant axon. So we can use this squid giant axon to demonstrate the passive electrical properties of cells. So largest axons of a common squid may be 2 centimeters long. Because these axons are so large, it's relatively easy to cut out a length of axon, or we can say the close up or ligate at the ends and penetrate the isolated axon with a microelectrode. So the microelectrode consists of a glass capillary that has been heated and pulled to a fine tip so that it can penetrate the cell membrane without causing damage. The capillary is filled with a solution of strong electrolyte, for example 3 molar potassium chloride, to minimize its electrical resistance. So when the recording microelectrode is advanced just past the axon membrane into the cytoplasm, like here you can see in the picture B, this potential difference across the axon membrane is the resting membrane potential. So when this recording microelectrode is advanced past the axon membrane into the cytoplasm, the voltmeter records the potential difference. So this potential difference is actually the resting membrane potential. So for all known cells, the polarity of the resting membrane potential is inside negative. That the inner membrane surface is negative with respect to the outer membrane surface. So this example actually reveals that axon's membrane resistance to current flow because without such resistance, ions would freely diffuse across the membrane and it would not be able to maintain this potential difference. So what would happen if the resting membrane potential, if we insert a second microelectrode into the squid and generate a pulse of electric current between it and the extracellular electrode? So what happens is that the current impulse would either depolarize or hyperpolarize the membrane depending on the direction of the current. So depolarization is actually the decrease in absolute value of membrane potential towards zero, and hyperpolarization is increase in absolute value of membrane potential away from zero. So recording the resting of membrane potential, we already talked about that. So we have one more, the selective permeability of a membrane that gives rise to a membrane potential. How this happens? So our model of passive electrical properties of cells describes that inside negative resting membrane potential maintained by all living cells. So what happens is that a cell's membrane permeability to different kinds of ions in the mechanism is the mechanism that established and maintains this voltage. So dissolved ions have charges and attract the polar water molecules around them. So the charged ions cannot mix with the non-polar tails of the lipid molecules in the center of this membrane. So how does a membrane's selective permeability to ions produce a membrane potential? Let's consider a simplified cell, an example, that contains a solution of potassium ions and non-permeating anions that are represented by the A. So we are concerned with its charge. The cell is in a bath of two non-permeating ions, that's the sodium positive and A negative ions. So we stipulate that the cell membrane is permeable only to K positive. So when the cell is placed in the bath, what happens is that these K positive tend to diffuse out of the cell down its concentration gradient. So because these charge neutrality is always maintained in the bulk solution, so A negative would tend to follow the K positive across the membrane. However, the membrane is not permeable to A negative. So therefore we can say the A negative tends to accumulate at the inner surface of the membrane, while K positive tends to accumulate at the outer surface. So the net charge on the inner surface of membrane and net positive charge outside tend to move these K positive ions back into the cell by forces of attraction and repulsion. So we can say that this system now has reached the electrochemical equilibrium in which there is no net movement of ions and no work is done. So for this system to come to equilibrium, there must be an electrical force, that's a membrane potential across the membrane. So that was the selective permeability of a membrane, how it gives rise to a membrane potential. So we have the non-spiking neurons that do not generate the action potentials. So ion concentration different result from the active ion transport and the passive diffusion. So we have different variations in the ionic mechanism of these excitable cells. As the ionic mechanism of, so how the universal or these ionic mechanisms of these action potentials. Studies have shown that basic concept of impulse generation that's elucidated in squid axons apply to most excitable cells. So what actually happens is that the action potential of vertebrate and invertebrate unmyelinated axons, the amphibian myelinated axons and vertebrate skeletal twitch muscles fibers have ionic mechanisms that qualitatively similar to those of squid axons. So we can say that ion channels from algae have been shown to function in mammalian neurons. So there are graded potentials in the non-spiking neurons. So what actually happens happens is that not all nerve cells generate these action potentials. So researchers report that increasingly numerous examples of non-spiking neurons, that's the neurons that do not generate the sharp spikes of action potential, they are called as non-spiking neurons. So these non-spiking neurons, they produce only graded membrane potential changes in response to a stimulus or synaptic input because they substantially lack that voltage gated sodium channel. So like here in this figure, you can see that the figure, it compares the signal transmission in spiking and non-spiking neurons. So what actually happens is that the input of both neurons, sensory stimulus or synaptic input, is graded in amplitudes, and so there is their output. So spiking neurons encode the graded input signals into trains of action potentials for long range transmission. The action potentials are recorded at the terminal to control that graded release of neurotransmitter. So what happens is that non-spiking neurons, in contrast, they are typically compact cells with short axons or no axons. So a graded potential change at one part of the cell can spread passively to the terminal without major decrement. So inputs and outputs of these spiking and non-spiking neurons are the same, but the short axon non-spiking neuron does not require spike encoding to carry out the signal over large distances. So that was all about the non-spiking neurons that do not generate these action potentials. So this is a mouse that was prepared for an optogenetic environment. We all know that human brain, which is a remarkably complex organ with billions of neurons and trillions of synapses, they communicate through timely electrical signals. So unexpectedly, the technology was possible because of light-sensitive transporter proteins and ion channel proteins that were first discovered in microorganisms a few decades ago. Optogenetics involves this taking the genes and coding these light-sensitive transporter and channel proteins. So inserting them into target cells and then delivering light to those cells in a way of controlling their functions. For example, the neurons in the mammalian brain, they can be targeted to express the light-sensitive channels. Then by delivering the light to those neurons, investigators are able to exert millisecond control over neuronal firing patterns, or we can say studying light. So cardiac muscle fiber and action potential, what does that mean? The action potentials of heart muscle fibers differ from the action potential generation in squamous axons in another way, and also their duration. So like here in this figure, you can see that the muscle fibers have action potentials with typical duration of like 100 to 500 milliseconds. So the long duration of cardiac muscle action potential is important because the action potential depolarization is necessary stimulus for myocardial contractions, and because cardiac muscles must contract for about 100 milliseconds to pump blood effectively. Or we can say a cardiac muscle fiber action potential has a rapid upstroke and a rapid initial recovery. So two separate inward currents and underlie the cardiac action potential. The first is fast current resulting from increased permeability to any positive ions, and you can see here in the picture, and in the picture B here, you can see the initial rising phase of the action potential that results from an increase in permeability to the sodium. So this line here, it shows a slow increase in permeability to calcium that maintains the plateau depolarization. And here you can see the other line, the other graph here. It indicates the permeability of potassium is decreased during the cardiac action potential. Or we can say the There are some other factors also that sustain the plateau, a decrease during the plateau in permeability to K positive from the resting level. So let's talk about the propagation of an action potential. So an action potential at one locus of an axon depolarizes an adjacent locus by setting up local circuits of current flow, like here you can see in the picture. So what actually happens is that a complete local circuit is a series of ionic and capacitive currents, which can be broken down into four components, like here in the figure 3 you can see. So these four components, 1, 2, 3, and 4, what are the different functions they perform? So at number 1, the locus of the action potential and ionic current begins with inflow of sodium ions through open sodium channels. So at the point 2, what happens is actually the ions flow in intracellular fluid, carrying the current to more distant parts of the membrane. And then at point 3, at the membrane here, which is called the capacitive current, what happens, the ion movements change the distribution of charges on the membrane capacitance. So cations accumulate along the membrane interior, displacing the negative charges and repelling an equivalent number of cations from the membrane exterior. So moving on to the fourth part is the extracellular. Here you can see an extracellular ionic current that completes the local circuit as cations move towards the locus of the action potential and anions move away. So the velocity of nerve impulse conduction increases with increasing axon diameter. How this happens, like here in this figure, it shows that the conduction velocity empirical increases with increasing axon diameters. So the data that are plotted on log coordinates, on these coordinates, the allometric relation, that's V is directly proportional to D, will plot as a straight line regardless of the value of D. The two lines that are forming a V at the center show the slopes for V that's proportional to D. So as you can see, the empirical plots in the red lines have slopes between these extremes. For some myelinated axons, V and D follow the square root relationship, but many myelinated axons have a more nearly proportional relationship of velocity to diameter. So myelin electrically insulates the major part of the axon, that's the region between the nodes or internodes. Here you can see that leaving only the nodes of the reindeer that we have of flow across the axon occur only at the nodes of reindeer. So in contrast to the continuous sweep of action potential over an unmyelinated axon, myelinated axon action potential jumps from node to node without active propagation in the internode. So these active action potentials are typically initiated at the axon initial segment, and then they saltate from node to node because these are the only regions in high concentration of voltage gated sodium positive channels. So that was all about the chapter 12, that was about the neurons. Now we still have 30 minutes, so we can complete the chapter 14. Let's move on to the chapter 14. That's the sensory processes we will be talking about. So in this chapter, we will be talking about the sensory processes that let's start with the example of the case study, a nocturnal encounter between a moth and a hunting bat. So sensory systems, they provide animals with all of the information that they have about their external environment, as well as most of the information they have about their internal environment. Let's consider an example consider the nocturnal encounter between a hunting bat and a moth. In the picture here, you can see the bat actually finds its prey by echolocation. It emits ultrasonic sounds at frequencies above the limit of human hearing and hears the echoes of its cries that bounce off the objects in the environment, rather like human detection of submarines by sonar. So moths, however, have evolved the auditory organs that are very sensitive to a bat cries. So a moth will fly away from faint bat cries. But in response to louder cries, the moth will either take erratic and apparently random evasive action or forward dive to the ground. So it's clear that it's of selective advantage for bats to have evolved auditory systems that allow them to use echolocation to detect and capture insects in the dark. So while also detecting and avoiding obstacles. So likewise, it's of selective advantage for moths also to detect and evade hunting bats. So these actually processes, these are the sensory processes, different sensory processes that help that moth to protect himself from hunting bats. So we will examine aspects of these auditory sensory system in both moths and bats in our next chapter. So along with other sensory systems that provide animals with information about their environment. So bats actually echo locate to orient and to capture insects, and moths have evolved such kind of auditory systems that help them to evade bat predators. So in this case, a moth hears the bat's cries and is able to evade the bat by diving towards the ground. So sensory system may be energetically expensive, but the information they provide is really crucial for the survival of moths, like here in this case study. So we have certain sensory organization of these sensory systems. So sensory receptor cells normally function as parts of a large system rather than in isolation. So basic function of sensory receptor cell is to convert stimulus energy into an electrical signal, which is called as sensory transduction. So these sensory transduction requires the specialized molecules which are called sensory receptor molecules. Like here in this table, you can see that sensory receptor cells can be classified into 4 different ways. So we have the oldest way of classifying sensory receptor cell is the sensory modality. As table here shows that however the animals perceive many other stimuli because these classical 5 senses. So humans are aware of additional sensory modalities such as balance and temperature, and there are many other modalities. So subject to Aristotle distinguished the 5 primary senses like vision, hearing, touch, smell, and taste. So these are the different kinds of sensory stimulus senses. That's the hearing, touch, smell, vision, and taste. So like here in this table, you can see that many animals appear to possess the receptive cells sensitive to modalities and qualities of stimuli not sensed by the humans. So second classification here is based on the form of stimulus that's perceived. So this is the classification of these sensory receptors or sensory modality based on the stimulus perceived. And here is one more classification that's based on the mechanism of the transduction. So in this we have 2 kinds of transductions. One is the ionotropic transduction and the other is the metabotropic transduction. So ionotropic transduction is received, sensory stimulus is received and then transduced into an electrical signal. A depolarization produced by the increased membrane permeability to cations resulting in cation, that's the sodium positive entry into the cell. All forms of mechanoreception, including hearing, seem to be ionotropic. So thermoreception, or we can say electroreception, and some forms of taste receptions, they only use ionotropic conditions. Other sensory systems, they always use the metabotropic transition, meaning the sensory receptor molecule acts like a neurotransmitter or hormone activated GPCR that helps in activating a metabotropic cascade. So finally, the receptor cells can be classified according to location of the source of stimulus energy also. We have the extroceptors, or we have the interoceptors. The extroceptors are sensory cells that respond to stimuli outside the body. Interoceptors respond to internal stimuli such as the pH or the osmotic concentration of the blood. So here's the principle of label lines in the sensory systems we have. Sensory receptor cells have two functional roles. One is the transduces some form of stimulus energy, converting it into an electrical signal, that's the receptor potential. And the receptor potential is a depolarization of this. As part of the encoding of sensory information, keeping the wiring straight by maintaining the central nervous system, the sensory modality or quality of sensation associated with the stimulus depends mostly on receptor cells that are stimulated rather than how they are stimulated. So figure here in this figure, it illustrates the generalization known as the principle of label lines. So most of the sensory receptor cells are quite specific in their kind of stimulus in which they respond. But many forms of stimulus energy may excite a receptor cell if there is enough of it. For example, any stimulus that excites the photoreceptors is perceived by light, whether the stimulus is actually light or poke in the eye or electrical stimulation of the optic nerve. So another key aspect of keeping the writing straight is that for each kind of receptor cell, the afferent axons, they typically maintain a geometric arrangement within the central nervous system. So insect cuticular mechanoreception, how this occurs. So insects have a hard exoskeleton covered with sensory bristles or hairs, each of which is a miniature sense organ called a sensillum, or we can say sensilla. So many sensilla are mechanosensory, whereas there are other predominantly chemosensory receptors. Mechanosensory bristles are hollow and contain sensory neuron endings. Here in this picture, you can see the bristle shaft or hair extends from the body of the insect as a part of its exoskeleton. So the very tip of the mechanoreceptor cell dendrite is attached to the inside of the bristle shaft at its base. And when the bristle is moved, it deforms the membrane at the distal tip of mechanoreceptor dendrite. So the receptor potential actually, if superthreshold depolarizes the sensory neuron enough to generate these action potentials, which often propagate to the central nervous system. So magnitude of the stimulus is encoded by the sensory receptor cells. Like here in this figure B, you can see that the stronger the stimulus deflecting the bristle, the greater the receptor of potential will be there. Depolarization, and the higher the frequency of action potential in the sensory neurons. Central nervous system then receives a coded message when it decodes from the train of action potential. So frequency of action potential denotes actually the strength of the stimulus. So here are some of the stretch activated channels. So further information about the stretch activated channels underlying mechanoreceptor comes from molecular genetic studies. So the channel protein structure is shown in this figure B. Here you can see that the structure is somewhat similar to that of a voltage gated K positive channel, or one of the domains of a voltage gated sodium positive So with six transmembrane sequences and a P loop that lines the channel with four. The channel is not actually the voltage gated, however, it's a member of a different but related channel family called the TRP, or we can say the transient receptor potential. So table here shows that the knowledge gives us some knowledge of the major transduction channels in sensory receptor cells. So these TRP channels in mechanoreceptors tend to have many encrine repeats, a protein structural motif that is used to link the protein to elements. This linkage allows the channels to open in response to stretch, enabling the cations to flow through the channels to produce a depolarizing receptor potential. So here are the hair cells of the vertebrate acoustico-lateraceous system, how it works. So these vertebrate hair cells are used in hearing and vestibular sense. So hair cells are the sensory mechanoreceptor cells in the vertebrate acousto-laterous system, which includes the vestibular organ. So these actually the lateral line system of surface receptors in fish and amphibians flow as well as and the mammalian cochlea and auditory organ that we will discuss later. So a vertebrate hair cell is an epithelial cell with, like all the epithelial cells, it has an apical surface that faces on underlying lumen and a basal surface that faces underlying tissue. So hair cells of acoustico-lateraceous system all work in the same basic way. The bending of this is transduced into the receptor potential. So let me give you some idea about the anatomy of the mammalian ear. So vertebrate vascular organs are the sensory function of acceleration and balance. For example, let's take a look at the anatomy of the mammalian ear. So the paired vestibular organs lie adjacent to the auditory organs in the inner ear and use similar hair cells. So they are called the vestibular because their hair cells project into fluid-filled chambers of any tiny rooms. So because of their complex geometry, the vestibular chambers and the neighboring chambers of the cochlea are collectively termed as labyrinth. Like figure here, it shows the structure of a human ear. You can see the external ear consists of pinna and air-filled externally auditory canal. The eardrum separates the external ear from the middle ear, which is also air-filled and contains the bones that transmit the sound vibrations from eardrum to the inner ear. So here in this figure B, you can see it shows the inner ear, including the vestibular organs and cochlea. So each vestibular organ comprises three semicircular canals which detect angular acceleration of head and body, and two otolith organs called the sacculus and utriculus. So figure here, the third picture here, you can see the orientation of semicircular canals. It shows that this conformation allows detection of angular movement in all three axes in space. So at the base of each canal, a region called ampulla contains a cluster of hair cells in a structure called the crista ampullaris. Here you can see. These are the crista ampullaris. So that was a brief about the anatomy of mammalian ear. So Drosophila tastes sensilium and its responses to different stimulatory actions, to stimulatory solutions. Insect taste receptor cells are located within bristles or sensilla. Similar in their anatomy to bristle mechanosensory sensilium, within this shaft of a taste sensilium there are dendrites of 2 to 4 chemoreceptor cells and often a single mechanoreceptor. So the shaft of the sensilium has one or more tiny holes that tip through which the water and taste molecules can enter. So in flies such as blowfly, these sensilla are located in the tarsus, the terminal segment of the leg, and the labellum, the tip of the extensible or mouth used for feeding. So a fly detects food in part by stepping on it. Stimulation of even a single tarsal sensory sensorium with a sugar solution, the extension of the proboscis. So further stimulation shows the responses of the chemoreceptor cells in tarsal taste sensorium to different stimuli. So talking about mammalian taste buds, so vertebrate taste receptor cells or epithelial sensory cells that synapse onto terminals of cranial sensory neurons, which then carry the signals to taste the centers in the brain. So taste cells are grouped together on tongue and back of the mouth in taste buds. So in mammals, the taste buds of the tongue are confined to small swellings, which are known as papillae. These are the foliate papillae, the fungiform papillae, and then we have the circumallate papillae. So all taste buds have a similar structure. Each includes like 50 to 150 cylinder elongate cells of at least 4 types that differ in morphology and staining properties. So until recently, investigators showed that only one of these cell types, that's the type 3rd, was a mature taste receptor because it was the only type that may define synapses. So simplest transduction scheme for salty taste is shown in the figure. Here the taste receptor cells have channels that are permeable to sodium positive. So when the sodium positive ion concentration within the mouth increases, the membrane potential of a salt receptor cell depolarizes. So the reason can be seen from any increase in external concentration of permeable sodium positive ions will cause a positive charge in the membrane potential. So talking about the olfaction, some insects such as male moths have elaborate olfactory systems. Each pinnate antennae of a male hawk moth contains an estimated of 360,000 olfactory receptor cells, and each antennae of a male polyphemus moth has about 260,000. So such members present a substantial fraction of the neurons in the insect's nervous system and represent a large energy investment. So the sensilla on the antennae of insects detect chemicals at a distance that are similar in structure to the taste sensilla. Instead of one or few small holes at the tip of the sensilium shaft, however, the entire shaft length is perforated by numerous tiny pores, only 10 nanometers in diameter. So here is a hypothesis that's showing the evolution of eyes. The photoreceptor cells and eyes of different groups have evolved similarities and differences. So what do we mean by photoreception? Is the response of a sensory cell to light that arose early in evolution. So all organisms actually detect light by using a pigment that's called the photopigment that absorbs the light. So photoreceptor cells are subdivided into ciliary and rhabdomeric. In ciliary photoreceptor cells, modified cilia contain the rhodopsin molecules, while as in rhabdomeric photoreceptor cells have collections of microvilli that increase the membrane of surface area. So most animal phyla contain examples of eye light responsive sense organs that differ in type and complexity. Eyes, as noted earlier, are the sense organs that contain the photoreceptors that are mediating these light responses. So we have two kinds of image forming eyes, in a camera eye and a compound eye. In a camera eye, a lens form and here you can see the camera eye, a lens form an inverted image on an array of photoreceptors at the back of the eye. In a compound eye, many facets called as ommatidia, each with its own lens together produce what's called a mosaic image. which conveys the information about one part of visual world, and the animal's nervous system constructs the image as a mosaic of tiles of individual uh, omatirial responses. So evolution of eyes is actually a subject of considerable speculation. There is evidence that most eyes evolved from a common ancestor, despite many differences in organization of different eyes. So, as we have already stated that all eyes acquire the same kind of photopigment, rhodopsin. So, moreover, disparate eyes share the homologous genes that regulate eye development. So, we have a single gene, PAX6, that initiate eye development in diverse mammals. Here you can see in the picture. And then similarly with other regulatory genes and eye development of many phylogenetic groups of animals. So it's unlikely that basic genetic plans for eye development and function that have evolved very clearly, but specific eye lineages, for example, camera eyes of cephalopods, cicads, or octopus and of vertebrates evolved separately using parallel expressions of these developmental and functional modules. So rhodopsin actually consists of the retinal conjugated opsin G protein coupled receptor. So rhodopsin is actually a photopigment that's composed of two parts, that's the retinal and opsin. So opsin is a G protein that's coupled with the receptor. So chromophore of animal photoreceptors is retinal, which is bound to the integral membrane protein called opsin to produce the light sensitive pigment rhodopsin. So figure here shows the chemical structure of retinol, which is the aldehyde of vitamin A, the form of retinol used in vision in most animals, including all the mammals. So in all species so far investigated, the aldehyde end of retinol is covalently bonded to the amino group of a lysine of opsin protein. So this bond actually buries the chromophore deep within the middle of rhodopsin molecule. So the structure of rhodopsin, as you can see here in the picture B, shows the 7 transmembrane alpha helix characteristics of all these GCPRs and the position of the bound retinol. So rhodopsin activates actually a G protein signal transduction cascade, or we can say GPCR, like other members of GPCR superfamily. So details of this can differ in different kinds of animals and sometimes even in different photoreceptors of the same animal. So that was all about the rhodopsin opsin. So this is the structure of mammalian eye and retina. So structure of the mammalian eye and retina, how it focuses on this photoreception. So retina of vertebrate eye is a developmental outgrowth of brain. It contains rods and cones and photoreceptor cells, as well as a network of neurons, that horizontal cells, bipolar cells, and ganglion cells, which actually perform the first stages of visual integration. So a pigmented epithelium also lies at the back of the retina. It absorbs the light that's not captured by the photoreceptors and performs many important metabolic functions, including the control of ionic environment around the rods and cones and synthesis of cis retinol. So retina is said to be inverted with the photoreceptors in the outermost layer away from incoming light. So that was all about the structure of the mammalian eye. So retinal pathways, they determine the receptive field properties of retinal ganglion cells. For example, we have off-center bipolar cell that hyperpolarizes an off-center ganglion cell inhibiting any generation of action plans. We have an on-center bipolar cell that depolarizes the on-center ganglion cells, exciting it and increasing its probability of generating action potentials. We also have the light in the surround hyperpolarizes an on-center bipolar cell which inhibits an on-center ganglion cell. So that was all about the sensory processes. Thank you.